Hi, everyone. I'm Christy Ballai. I am one of the co-PIs of the Insect Decline Research Coordination Network, and welcome to our monthly seminar series. So today we're going to do sort of our typical path of having a medium length talk and then a longer discussion after the fact. Today we are joined by Bashira Chowdhury. Bashira is the science coordinator for American Forests' Texas program. But prior to this role, she was an ecologist with the Department of Entomology at Mississippi State. So she worked on various plant and insect conservation projects when she was there. But while she was there, she developed what she now refers to as her side hustle, her deep interest in entomological history and using historical records to tell stories about how insect populations play out over time. So she partnered with Jim Geeson, an associate professor of history and president-elect of the Agricultural History Society, almost a horticultural society, um, to study insecticide use and biocontrol using archives during the beginning of the National Boll Weevil campaign. So their Boll Weevil work is ongoing, and so Bashir is going to tell us a bit about it today.
Thank you, Christy, and thank you again for having me. And I'm going to preface this, and I know I'm talking to conservation biologists and entomologists and ecologists, and I have no fancy graphs or fancy graphics or anything. This is very much grounded in history. And so there's going to be a lot of words, and I'm going to be telling you a story. Um, and first off, I really want to frame this story in that I want to introduce you to insects that are increasing, not decreasing, but still need to be conserved. I know this is very ironic given that I'm talking to a NSF coordination network on insect decline. But there are insects that are increasing that still need conservation. And so I just wanted to frame our talk and discussion today with that. And before we really kind of get too far, I really want to explain the boll weevil eradication campaign, especially to those that are not maybe not from the South, not familiar with cotton. The boll weevil is a small weevil. It's on the left of your screen. And from 1978 to 2003, the USDA and state governments and land grant universities waged a war on this tiny little weevil in mostly southern cotton fields. And I apologize for the poor Google map, but most of this work happens in the Southeast because that's where we grow cotton. So there was some eradication efforts both in Arizona and California, but truly we grow most of our cotton in the Southeast, in the southern states. And so this war really was waged starting in 1978 in North Carolina and extended all the way to South Texas. For the most part, this campaign is considered the most successful eradication campaign in US history. It ended in 2003. And the only holdout for boll weevils is in the state of Texas, along the Rio Grande border, where boll weevils are native. Um, this campaign largely involves spring malthion during the late summer and early fall to primarily target overwintering boll weevils. And they would also follow up with trapping in the spring. And this just is a really bad slide that just sort of summarizes really what the campaign was about. A lot of the campaign was really grounded in a lot of research that happened in the 1950s and 60s. For those of you familiar with entomological history, Edward Nippling is considered one of the key architects of this campaign, and he was a very prominent USDA entomologist in the 50s through the 80s. And in the late 50s, he was a part of a team that discovered a lot of different characteristics about boll weevil biology, particularly that they enter diapause going into the winter. And so this campaign was really targeting those adult boll weevils that were about to go into diapause because they really wanted to prevent the following season's population of boll weevils. So there was intensive spraying of malthion. And then, likewise, much later in the 60s, they developed pheromone based traps in order to both catch male boll weevils, but also to assess whether or not they had fully eradicated boll weevils from the field. And finally, they specifically used malthion because boll weevils were susceptible to malthion. And the boll weevils, for the most part, had already been resistant to DDT at this point. Parathion was considered very hazardous to work with at the time. And so they ended up going with malthion. And but collectively, these breakthroughs really catalyzed this campaign to go forward. And outside of just this legacy of a very, very successful eradication campaign, campaigns leave a lot of traces and that they leave a lot of paper. And so they left dozens of boxes of field data and hundreds of government documents, and also thousands of specimens that really chronicle these different moments in the eradication effort. There are collections scattered across land grant universities, all the way from North Carolina State University to Texas A&M to Mississippi State, which was the actual epicenter of the boll weevil eradication campaign. But today's talk is actually not about boll weevils. As I mentioned at the beginning, it's about insects that are increasing, not decreasing. And it's really about these guys. It's really about the survivors. And these are the insects that actually survived through these multiple rounds of malthion. You find them latent in all these old field notebooks and USDA bulletins and in pin specimens that are tucked away in museum drawers from Raleigh to Texas A&M to Starkville, Mississippi. And in particular, you actually find a lot of those middle tier insects. So a lot of your natural enemies. So your nabbids, your carabids, and not necessarily your insects, but also your wolf spiders that actually survive these repeated onslaughts of spraying. And I know for a lot of you out there, you assume that natural enemies are highly susceptible to insecticides. And that assumption is one of the core principles of integrated pest management. And that is definitely true. And I know some of you might be IPM-based entomologists, and you definitely encourage your farmers to delay how they spray. But however, the legacy data, particularly from the boll weevil campaign, shows that a lot of these insect predators persisted through this onslaught. And this talk is really about these survivors and their resilience and how we can find them and conserve them. So, really, finding survivors involves a timeline. So, as I said, this is history. History always requires a timeline. So, I want to give you a sense of how the campaign actually unfolded. So, in basically from the early 50s through the 1960s, there was this talk of eradication, particularly because you had these major research developments in the boll weevil campaign, in boll weevil biology. So particularly, again, Edward Nippling is a key figure in that he formally proposes to the National Cotton Council that we eradicate boll weevils. And then a huge breakthrough for boll weevil folks is in 1962, Congress, the US Congress, gave basically Mississippi State the money to start the USDA National Boll Weevil Lab. And then another huge milestone is 1969. After Nippling and others in the National Cotton Council lobby successfully to the United States Congressional Committee on Agricultural Appropriations um, for funding for a pilot eradication experiment, which happened to be in Mississippi. And that both the national lab and the eradication experiment happened in Mississippi, not by chance, and not because Mississippi necessarily is one of the best places for for entomology, but in part because the head of the U.S. Congressional Committee for Appropriations, Agricultural Appropriations, was Jamie Whitten, who was a very powerful Mississippi Democrat at the time. So it's one of the reasons why this story very much revolves around Mississippi. And the pilot experiment runs from 1969 to 1973. They are unable to successfully show that they were able to eradicate boll weevils. However, they were able to convince Congress to slip in boll weevil eradication into the 1973 Farm Bill, but Congress did not allot any funding for it. Then a couple years later, in 1978, funding comes along and they start the eradication effort in North Carolina and Virginia. And then finally, by 2003, the United States is declared rid of the boll weevil, except again for South Texas. So, and this is again along the Rio Grande border. Now, this timeline, particularly when you're thinking in history, runs concurrent to other timelines. And this is why, and understanding these other timelines is very critical to finding these survivors, because we have to place these documents in terms of these other timelines. So this is also a very, very famous period in the environmental movement. The eradication campaign follows on the heels of both Rachel Carson publishing Silent Spring, which was a monumental piece in the environmental movement. It did spark a lot of public outrage that eventually led to DDT being banned and the EPA being founded in 1972. And while this environmental movement didn't necessarily prevent eradication from using malthion in heavy doses, it definitely shaped how it used malthion to some extent, and it also influenced and made aware that the scientists needed to think about other things than the boll weevil. So you actually start seeing in some of these documents, particularly in the 1970s, the discussion of non-target insects, because before then, boll weevil documents really don't mention them. So again, it's very critical. If you're going to find the survivors, they've got to be mentioned in the written record. And we start to really see that in the 1970s. And then of course, there's another timeline that many of you are going to be much more familiar with. And that is the timeline of integrated pest management and biocontrol. And I placed it separate to the environmental movement because it was very separate. Integrated pest management and biocontrol, while it seems like there's quite a lot of influence from the environmental movement, and certainly there is, it had its own evolution that ran parallel to the environmental movement and to some degree the eradication effort. And first, it really starts with this increase in DDT resistance on farms that was really observed in the 50s and 60s. And before then, natural enemies was sort of out of vogue at this point. For those of you that are very well versed in entomological history, you might remember in the late 1800s, early 1900s, the cottony cushion scale out in California and the import of the Vidalia beetle and this big push by the USDA to start importing all these natural enemies. But by the time DDT arrives after World War II, natural enemies were very much out of vogue. There was still just a small smattering of entomologists around the country working on it, but really people were relying on, entomologists were very much relying on insecticides. But with the increase in resistance, you actually started to see a resurgence in biocontrol research. This ultimately led to Carl Huffaker, the famous uh, biocontrol expert, being awarded millions of dollars in NSF funds to establish a formal research program for biocontrol for 6 commodity crops. And that happens coincidentally in 1972. Richard Nixon was actually the one who pushed for it at the same time he was founding the EPA. Much later in 1978 and into the early 1980s, there was formal federal funding for integrated pest management as we see it today. So those are sort of the timelines that that coincided with the Boll Weevil eradication campaign. Now, um, really, where the natural enemies start to pop up, and I'm giving you a more concentrated look at this, is we really have these 3 piles. When it comes to history, it's not a perfect record. You have papers, and there's lots and lots of papers in the archive, but really you can kind of lump them into 3 piles because it's really just what people happen to leave behind. So we happen to have a lot of papers and specimens and legacy data related to the pilot campaign that was in southern Mississippi from 1969 to 1973. Then we also have a second pile, which is all about the Huffaker Project. So Carl Huffaker made it to the Mississippi Delta between 1974 and 1976. And in 1978 to 1981, IPM and Eradication meet in Panola County, which is basically the center of Mississippi. And again, I just wanted you to still keep in mind that our timelines, our parallel timelines of both the environmental movement and IPM are still ongoing at this time in influencing these piles. But these 2 timelines really also help to bring out why we start to see our survivors within these piles and how we identify them and understand their unnatural history. So first, the pilot eradication campaign was a 4-year effort from 1969 to 1973 to demonstrate that you could eradicate the boll weevil. So they just wanted to show that you could eradicate boll weevils from an area that there was chronic infestations. So basically they established a core eradication area and had a buffer zone, and they sprayed malathion on cotton and the surrounding woodlands. And they started to measure natural enemies. This is where our survivors show up. It's not to say there weren't survivors before, but when it comes to history, we've got to have a record. We've got to have a written record. And this is where we first start to see them. And so at this time, they had identified Geochris punctypes, and they were tracking it. This is, again, the first time we really start to see them tracking it. And they start measuring it at the beginning in the spring. There's about 300 Geochris per acre, and it goes down to 6 Geochris per acre by the time they really start intensively spraying malathion for adult boll weevils. Then the second big time we see natural enemies is in the Huffaker Project. And granted, Huffaker was all about natural enemies. So of course you had to address it. They had to go find it. But in particular, they were looking for natural enemies that were resistant to hard insecticides. And that quote actually comes from the NSF Progress Report in 1975. They were specifically looking for natural enemies that were resistant to hard insecticides. And correspondingly, they went out every single time after they sprayed malathion looking for natural enemies like navids and Geocaris. And coincidentally, the Mississippi State Entomology Collection, the majority of their navids come from the Huffaker Project during this time. There's about 4,000 of them, and they come from this time. And then finally is the optimum pest management trial. And this is the stack of notebooks that used to sit on my desk at Mississippi State, where they tried to now systematically document natural enemies across multiple microhabitats within a cotton field, everywhere from the soil to the plant itself. And they documented no impact of the eradication effort, again, this very intensive spraying of malathion, on natural enemies communities. And in particular, they were looking for, they did everything. First of all, they pitfall trapped, they devac, they aerial netted, and they looked, in this case, they looked more widely for wolf spiders and statistics that you all would call exceptionally rudimentary. I'm not quite sure y'all would even call it statistics at this point because it's so basic, but they basically, their summary reports argued there was no impact to natural enemies. By this point, they didn't use the word resistant, but they argued that it was compatible. And that's what they really wanted to show, is they wanted to show that IPM and eradication could work hand in hand, that biocontrol was not affected at all. So collectively, all these documents and all these museum specimens tell us a story about how these natural enemies survived and then later thrived through these repeated malathion sprayings. And again, it's not necessarily the story that you all as ecologists would expect. Like, I don't have a pretty graph. I just have words across multiple, hundreds of documents over time. I've got, insect specimens that increase over time. And there's just this speculation that we start to see pesticide-resistant natural enemies increase over time, particularly natural enemies that are resistant to malathion. And it suggests that there's hope, that we do see through these intensive eradication campaigns that we're going to have insects make it. And you all are here working and thinking about insect decline. And so I wanted to leave you with that there is this potential that these insects are making it. But again, this is also a conservation problem, because even though the species are making it, their genes are not necessarily making it. So we can tell from the data that there is most likely very strong selection going on. So as I mentioned earlier, you go from 300 Geocaris in a field to 6. And I'm sure that rings alarm bells for many of you that there's got to be some sort of bottleneck, which means you're going to have both bottlenecks and likely in all these different farms, potential genetic differentiation. And so this is why this is a conservation issue, is that you do see these pesticide-resistant natural enemies coming through, but they're coming through at a cost to their genetic diversity. And that's the heart of conservation, is as conservation biologists, we seek to protect genetic diversity. We want to protect that variation in order to facilitate adaptation to the future, but also that is natural heritage in and of itself. And so, of course, our next step is really to identify conservation needs for pesticide-resistant natural enemies. And our small little crew of me, Jim, and science librarians, one of whom he's here today, are asking two big questions, both which are couched within conservation biology. The first is, who are these natural enemies? And really, this comes down to documenting biodiversity. We can't do conservation unless we know who on earth we're trying to save. And then second, how do insecticides actually threaten these insects? Another way of thinking about this is a threat analysis. So because this is history, but we're using history to do ecology. I just want to kind of walk you through our process and of how we're actually doing this. So when it comes to history, you build a corpus, and in our case, a digital corpus. And basically, that's a fancy way of saying you first identify the archives that you're going to be working with. So yes, there are a lot of different archives for the boll weevil, but you specifically, when you go into the archives, you actually identify the banker's box and the folders that you're going to be working with, because you're not going to scan everything. You're only digitizing the documents that are relevant to your work. Second is you annotate it. And there's no other way to say annotate it, but you're note taking along the side. It's similar to what you guys have been doing when you were undergrads, making notes on the side of a biology textbook. And then finally, you take your annotations and you actually visualize it. And so I want to break down our first question in that we're asking, who are pesticide-resistant natural enemies? So we're still sort of in the process of this, because this is actually quite a long process, and history takes a long time. The best way I can explain it is historians take sometimes 10 years to do their work, while y'all might take 10 months. We're trying to find a happier medium that is a little bit closer to the 10 months and not the 10 years. But really, right now, we're trying to finish up making digital copies of our reports, our legacy data from the pilot, the eradication, pilot eradication, Halfacre, and the optimal pest management trial. And then we have to mark up that language. And we're not marking everything up. Specifically, we're looking for language related to insecticide resistance, non-target insects, and biocontrol of secondary cotton pests. And there are various software packages that allow you to annotate text. But specifically, you're looking for those words. And I don't know how to explain this, because this is where that humanistic aspect comes in. But after reading these documents over and over and over, you have a good idea about what words you're looking for. There's no magic way to do it. There's no AI app that's going to help you here. It's just good old fashioned brain power. And this is where the history comes in. This is what the heart and soul of history comes in. And this is where it merges. When we mark those historical annotations, we now facilitate the ecology when we try to visualize those patterns. And specifically, we want to correlate insecticide resistance to natural enemies. And this is a little bit complicated in that insecticide resistance in and of itself has meaning that changes over time. So we also anticipate this is going to be a complex correlation. Our second question, again, is our threat analysis question. How do insecticides really threaten these insects? And so my co-conspirator here, Karen Burton, who actually is here today, she and I are right now digitizing copies of the legal proceedings from the eradication campaign in South Carolina. Yes, that is the legal proceedings. As any federal mandated campaign has with it, there were lawsuits. Farmers did not want to do this. So again, I'm mentioned briefly in 1973. This was federal law that the U.S. government was going to eradicate the boll weevil. So state governments complied and wrote it into their state constitutions. And if you look into state constitutions, state laws for every single state that was affected by the eradication campaign, there is a law that says that farmers within that state have to eradicate the boll weevil and comply with the campaign. Of course, a lot of people didn't want to do that. A lot of farmers fought to do that. And what's interesting about the legal proceedings is that in anything legal, you have to say what's going on. There's depositions that are taken. And in the depositions, the farmers actually say what they're doing. So you actually know what insecticides are going into which fields. And as a part of the campaign, and of course, again, it's a legal thing, they have to keep track of all these farms. And so Karen and I have found a lot of the legal proceedings for Anderson County, South Carolina. And in these legal proceedings, the farmers actually say what they're doing in their cotton fields at that time in those months because they legally have to. So currently we're digitizing those. She and I plan to mark up all the language around insecticide use and farmer practices. So we then plan to track the changes in insecticide use and farmer practices as the campaign continued. And in this case, the campaign reached South Carolina in the 1980s. And again, it's the real brunt of the history happens in the annotation and in the visualization. That's where the ecology, that's where the conservation happens. And then we're still in the process of doing this work. It takes a long time to do history. We've been doing this now for almost 3 years. And believe it or not, it took us quite a while to make sense of all this and to make sense of all these documents. And I just want to thank, first of all, my mentor. He's been through this the whole way, Jim Geeson over at Mississippi State. We have gone through so many rabbit holes together. Karen Burton is willing to follow me in every single rabbit hole. And we found dead ends, but we are now working on threat analysis together. And then finally, Brad Brazil, who is Karen's counterpart in Mississippi State, who actually helps us find all these things. Not everything is in an archive because university archives really, they want to have old stuff, and old things are considered like 1900s, not 1970s or 80s. So those don't end up in the archives. So that's why I have to work a lot of times with a science librarian to find those things that aren't yet archivable. So I just want to thank my science librarians. And again, I want to thank you all for listening to my story about the unnatural history of pesticide resistant insects during the Boll Weevil campaign.